You ready? Okay. Can you see me? All right. Good evening, everyone. My name is Margaret Angel, and I am the Head of Partnerships and Strategic Initiatives here at the Roux Institute. We're thrilled to have you all here live in person, and welcome to our guests who've joined virtually. It's a really wonderful chance for us to gather here for our regular speaker series. We call it Our People, Our Work, Our Community. And it's been an initiative of the spring after the Omicron surge to open up our campus and welcome everyone and introduce some of the key people and themes of the Roux Institute. And one of the great joys has been to set up these evenings to highlight the directors of research at the Roux Institute. And it is a real pleasure to be able to kick off that portion of this series with Ray Winslow. And so I just want to welcome everybody. I hope you enjoy the evening and encourage you to come to as many of these events as you can, and encourage anybody you know who's interested in these themes to attend. And I'm going to hand the mic over to Mike Pilastri, who leads all of our academic research and learning here at the Roux Institute, and he's going to do a proper introduction of Ray Winslow.
Thanks, Margaret. This is a great day. The last year and a half of building the beginnings of our research leadership team, culminating in an event like this is really exciting for me personally and professionally, and for the Roux Institute. We're really excited. So it's a pleasure to introduce Ray Winslow. So Ray joined Northeastern just about a year ago, as I would say. But before he came here, he was at Johns Hopkins University for 30 odd years. In fact, he was I want to make sure I get the name right. So the Rajanira Singh Professor of Biomedical Engineering, and the founding director of their Institute for Computational Medicine, essentially inventing the field. And so you could ask, why would he come to Maine? He gave up a endowed named professorship and tenure to join us here at the Roux Institute. And the answer is because Ray's a Mainer, and Ray loves Maine. And Ray was looking for an opportunity to apply his gift, his talents, his expertise, and attract talent to Maine to work in a field that he's so passionate about, which is applications of predictive analytics to healthcare. And where else? I mean, Ray has made some really remarkable connections between Jackson Labs, University of Maine, Maine Health, places that are going to prove to be really powerful collaborators where he can actually translate what he's doing into real healthcare, into life sciences and medicine research. So we're really excited to have him here. And so that's why he's here. Also, if you're around and you sort of wander the building later afterwards, you'll see a picture of Ray in his dry suit. I don't know if you know this, but he's quite a kayaker, a sea kayaker, and the hardiest person I think I've ever met. So anyway, it's a great pleasure to have Ray kick this off, and so enjoy. Ray.
Mike and Margaret, thank you. You can't believe how perfectly you set me up with that introduction, Mike. Thanks very much. I don't usually put pictures and seminar series titles in my intro slide. It's just the name of my talk and me, but I couldn't resist it here for a couple of reasons. And the first is the Roux Institute, Our People, Our Work, Our Community Seminar Series. I love that name. That's a fabulous name, Margaret, for the seminar series. Then there's this picture of Portland. And I was looking at this morning thinking, why did I put these two things together? Well, I know why. One of my favorite poets is Henry Wadsworth Longfellow. And he wrote a poem called My Lost Youth. The first line of that poem is, "Often I think of the beautiful town that is seated by the sea." And it goes on to talk about Portland, never naming Portland, but speaking of Portland. And I thought, there she is, our beautiful town that is seated by the sea. He was talking about his home. And to me, our people, our work, our community, that speaks of home. This is our home in the Roux Institute and in Portland and in Maine. And I am sure glad I'm here. Every day I'm delighted I'm here. Okay, let's begin. I have a strange title for this slide because I know that many of you have heard me say these things, frankly, ad nauseum, right? And I think there's a strategy there. I think having a short thing you say over and over and over cements an idea into people's heads. And I began saying it with students at Hopkins. I said these things all the time. So the first thing I said was, medicine is increasingly becoming a computational discipline. Every one of you who has heard me say this, I believe this is really true. And what we're building towards here is kind of the essence of computational medicine, how I define it. Well, I think this is why computational medicine is what it's doing. It's quantifying medicine. It's making it a computational discipline. Here's something else that I say. More recently, I've started saying, the algorithm would become a member of the healthcare team. Now, I kind of like this. We even wrote a paper on it, believe it or not. And a clinical journal actually published this paper on how the algorithm would become a member of the healthcare team. And so I love that phrase. I think it's right that algorithms and other kinds of caregivers are going to work together in the future, with algorithms making recommendations, not decisions, recommendations to those caregivers about how to deliver better care to their patients. So here we go with what we have over the many years boiled down to the essence of computational medicine. This slide title is The Emerging Discipline of Computational Medicine. I gave a talk last week with this title, and after I gave the talk, I said, Ray, stop it. Computational medicine isn't emerging anymore. It's emerged. There are now departments of computational medicine. UCLA created the first department of computational medicine. A student from the Institute for Computational Medicine was their first faculty hire. So it's happening. It's not emerging. It's here. So this is how we define it. In computational medicine, first thing we do is we develop computational models of disease. Models is a broad term that I'm using here. I don't mean it to refer specifically to AI or machine learning or anything else. It's any kind of mathematical formalization that enables us to understand really quantitatively what disease is and how disease processes function. So armed with these models, we figure out how to personalize them using patient data. It's an easy thing to say. It's rather hard to do. It can be rather hard to do. And so this is a non-trivial aspect of computational medicine. And of course, the third thing we do is we apply these models that are now personalized to that patient there to deliver better healthcare for that patient. We talked about it in this paper. This is not a technical paper. If you want to view our thinking, this was published in 2012, I think. That was our stake in the ground, telling the world this is what we think computational medicine is. So go take a look at it. So what I'm going to do now is I'm just going to immediately plunge into the word models and speak to you about how I see models in medicine. Okay, we're going to walk our way through this slide. This slide is about using machine learning to develop models of disease. I want to use it to explain my view of what machine learning is. It's really pretty simple. Let's hold in our minds the following example. So, in heart disease, people who are suffering from recurrent episodes of arrhythmia, a common treatment these days is to implant in their heart what's known as an implantable cardioverter defibrillator. This is a device that senses electrical activity of the heart and shocks the heart when that activity is sensed to keep these subjects from going into cardiac arrest. This is a non-trivial thing. ICDs deliver substantial amounts of energy. Patients can go unconscious when ICDs fire. Here's another fact. The firing rate is 3 to 4% a year. Some patients have ICDs placed and they never fire. These two pieces of data are saying we don't really understand who should be receiving an ICD and who shouldn't. So suppose we want to figure that out. We want to build a model that helps us answer the question, should this patient receive an ICD? Maybe we can collect a lot of data about patients whose ICDs don't ever fire or don't ever fire appropriately. All firings are adjudicated by physician panels to see if they were appropriate. And we collect information from patients whose ICDs fired appropriately. And suppose our task is we want to take these data and make a decision. Yes, this patient should receive an ICD. No, they shouldn't. That's the example I'm offering up to you here. And now let's work our way through it. So we're going to collect data from these patients. It could be from the electronic medical record, heart rate, lab test results, information on mobility, the nature of their heart disease, whatever that information is. We're going to collect it all together. These observations from each patient, I'm denoting by this vector X. That's a set of things we measure in a patient. We're going to separate this total data set into what's called a training set, S, and a test set called U. We're going to learn from the training set. We're going to try and understand how to relate the things that we measure in a patient to what I'm calling class labels, these things L sub I, the labels. For patient I, with these data, their label is, yes, I should receive an ICD, no, I shouldn't receive an ICD. That's the label. The machine learning problem, the supervised machine learning problem, I should say, is to take this set of features and the associated labels learned from data. So here's a feature from a patient whose ICD fired. That's the data, that's the label. Maybe here's a patient whose ICD has never fired. It's their data, that's their label. No, ICD didn't fire. And from this, we want to learn a function F that maps the data into a label, so that when we get a new set of data from a new patient, we have this function F that can tell us how to assign the label to that patient. Should they or should they not receive ICD placement? This is the supervised learning problem. And once we've learned this once we've learned the function F from the training data S, we test it on the held out data U. We've done no training. This is the cardinal rule of supervised machine learning. Never test on the training data. Hold it aside. This is new data that you've never seen before. How well does the function F generalize to information that it's never seen before? And that can tell us how well F works. So what are the strengths and weaknesses here? What are the pluses? Learning the function F doesn't require deep knowledge of mechanism. We don't have to understand in biophysical detail every aspect, necessarily, every aspect of the electromechanical functioning of the heart in order to build an F in this manner. We just need data and outcomes, and we learn this F. So it doesn't need deep knowledge of deep mechanism. And why does that matter? Because getting that deep biological knowledge is hard and slow. So learning F is relatively fast. And computing F, once we've learned it, is trivial. F is usually a really, really simple function. Boom, you give me data, boom, I've got the label. Those are the pluses. The minuses, it's back to the mechanism thing. The function F doesn't know anything about mechanism. It's very desirable to understand what's going on and not just operate on correlations and relationships. So at best, machine learning enables you to make kind of a weak inference about mechanism based on the features that are important to assigning a label. But it doesn't really inform you in a deep way about mechanism. And then finally, the most important thing is performance is influenced strongly by data quality and the quantity of training and test data. A lot to be said there, which I'm not going to say. I'm just going to go to the next slide now. So here's an example of, oops, I'm stepping out of the camera zone. I was told to stay between the plants, and I warned them that I would wander all over the place. Okay, so here I am between the plants. A lot of what we do in life sciences and medical research is predictive analytics. This illustrates the general notion of predictive analytics. There's a patient here, lots of equipment, data flowing from that patient. It could be the high-frequency waveforms like ECG or EEG or blood pressure waveforms. It could be minute-by-minute vital signs data, heart rate, respiratory rate, SpO2. It could be infrequently entered and irregularly entered healthcare data out of the electronic health record, but all of that data gets captured for study, and it gets analyzed by applications or apps that are performing this predictive analytics. What is the goal here? To quantify the state of that patient at this time and use machine learning to make advanced predictions about where that patient is headed. Are they getting better? Are they going to get worse? Are they going to get worse in a particular and serious way? If they are going to get worse, can we make that determination reliably, and can we feed information back to caregivers to help them treat this patient? And so here's a phrase I first heard from Oussama Fayed, humans in the loop. I think our very first meeting on Zoom when I joined here, maybe before I joined, humans in the loop. There are humans in this loop. There are caregivers, patients, and families in this loop for sure. Here's something I learned from Melanie Torrey. There she is, yes. The importance of understanding how to feed back these recommendations, not decisions, recommendations to caregivers and families in a way that they can harness them most meaningfully. So these are aspects of the loop that I've had filled out for me when I came to the Roux. I do want to say a few more things about this picture that's important and compelling. It was taken by my colleague, Jim Fackler, who's an attending physician in the pediatric ICU at Hopkins. There's a child here who has multiple organ dysfunction syndrome. They've undergone both heart and lung failure. And they are being sustained on what's called an ECMO machine, extracorporeal membrane oxygenation. This external device removes carbon dioxide from their blood, infuses the blood with oxygen, warms it, delivers it back to the child in an effort to rest the heart and the lungs to help them recover. It's the leading cause of death in pediatric ICUs. And what I think of when I see this picture is imagine being the family of this child and coming to see your child with all of this stuff around your child. It's incredibly traumatic. It's incredibly traumatic. That's why I say perhaps we can help. Maybe there is a way with our skills, our engineering skills, our understanding of data visualization and analytics, that we can help here. That's what we're going for. We want to be able to give caregivers advice that they will weigh along with everything else that they weigh in their decision making and help that child or the patient in general. Okay, this is a bit of technical detail, but I'm going to talk about it anyway. This illustrates the notion of a risk score. This particular illustration is every patient in this study has sepsis. They're in an adult ICU, they all have sepsis. We're trying to predict who will evolve into septic shock at some future time point. We want to build a risk score for that. There are principled ways of doing this, of building this risk. This number here is the probability, P for probability, that a patient with sepsis is going to evolve into septic shock. There are multiple machine learning methods for building and learning that function P. One of them is logistic regression. That's what I show here. You take data from the patient, put it into this function, you get an estimate for this probability P. The beta values here, they're parameters. You've got to learn them from the data. The beauty of logistic regression is optimal ways of doing this. You can learn the best set of betas for doing this for a function of this form. There's an optimal solution to this problem. You're guaranteed to converge to that solution. The more data you have, the closer your probability estimate is to the actual probability. It's an unbiased estimator. So this is really powerful, really cool. You can build these risk scores. That's what I'm illustrating here, evolving in time. This is time. That's the risk. This is an imaginary risk score in the blue. We're computing this probability of going into septic shock, given you're in sepsis. We hypothesize that you can define a threshold on that risk such that if you exceed that threshold, and at that time point say, this patient will go into shock, that you can do it reliably. So how do we find that threshold? This is the classifier. It's the world's most trivial classifier. It's a one-parameter classifier. Find the threshold. How do you do it? Take your training data, test every threshold. Find the best working threshold based on your considerations. Ask the question, is that good enough? Are the actual performance results that we see adequate? If yes, wonderful. And there is some early warning time. Remember, what I'm illustrating here is that this time the patient conforms to the clinical definition of septic shock. So this early warning time is the time between when they actually enter shock versus when we say they will evolve towards shock. Turns out on average in this problem, it's about 8 hours in advance. That's pretty good because we know in septic shock, patient mortality increases by 8% every hour. So it's a rapidly evolving disease. So 8 hours lead time to know that this is coming is pretty good. But this is how these problems work. We build these kinds of risk functions. So here's an example of results. Let's just focus on this panel. I should have just showed this panel because it takes us back to the ICU photo of a kid with multiple organ dysfunction. The problem that we're looking at here is, can we predict children who are admitted to the ICU who will develop multiple organ dysfunction, like the child in the original photo that I described to you? Can we predict who will go into multiple organ dysfunction? Well, we can do that. And here is an analysis that takes all of the risk scores together of all of the children out of the pediatric ICU at Hopkins over a 5-year period who evolved into multiple organ dysfunction syndrome, and it shows their risk. What's the big thing you see in these risk scores? What you see is that all of a sudden, in the blink of an eye, the risk is sky high. We see this in every single ICU problem that we've studied. There is a moment in time where risk shoots sky high. It's very, very clear here. It's clear here, this is the sepsis and septic shock transition. Nothing different until the moment of threshold crossing, and the rate of threshold crossing is very fast. Now here in this organ dysfunction problem, on average we can do it 37 hours in advance. For some kids it's 60 hours or 72 hours. For other kids it might be 20 hours or 15 hours. There's a distribution, but the median of that distribution is 37 hours, enough time to get in there and potentially help that child not go into multiple organ dysfunction. And what this means here is that there's two populations. That after threshold crossing, after we make the call, these patients separate into two groups, a high risk, high mortality group, a lower risk, lower mortality group. What is remarkable here is that in this study, we demonstrated that when we make the call, a child is in the high risk group, we get it right 97% of the time. We may not pick up every child in the high risk group. We pick up a great many of them, roughly 80% of them. But when we say this child is at really high risk, we are right 97% of the time. I think this algorithm should be in every pediatric ICU in the country. because I think with this positive predictive value, we will not be generating false alerts to caregivers. When we say that kid is headed towards MODS, we're right most of the time, and it's important to not over-alarm caregivers or they'll just blot you out. I mean, it's just an annoyance. If you're wrong most of the time, they will not take action. If you're right most of the time, our hope is they will take action. And one more final point, I think these abrupt transitions indicate that in many of these ICU problems, while we don't understand biologically what is happening, what I think about in my mental model of this, my intuition is that the body is able somehow to fight off the infection that is present in sepsis up to a point. Or up to a point, the body can resist what's happening that leads this child into multiple organ failure up to a point. But then there's a bifurcation. Our defense mechanisms just collapse and we become very, very sick. So this is marking in time, I think, it's telling us this is when an important biological event that we don't yet understand occurred. We should go look for it. We should go figure out what that event is. Use this as a clue. Okay, let's move on. There's more to say there. Okay, now I want to move to my second example of modeling. It's a very different kind. I hope I can make this intuitively clear. And I refer to this as dynamical systems modeling, or maybe mechanistic modeling is an equally good term. This is intended as a representation of a biological network, a molecular network where different molecular species are interacting with one another and connected to one another, and their dynamics is evolving over time. This is a signaling network. These are receptors in the cell surface. Here's a G protein coupled receptor. Maybe an agonist binds to that and causes the concentration of these various kinds of signaling molecules to vary in time. Dynamical systems modeling tries to capture and describe all of that, the mechanism of that. So if we think of the state variable X as being the concentrations of these different molecules in this pathway, and by the way, this pathway could subserve a disease. Its function could be perturbed in disease. So in general, we want to understand how this pathway works and how it gets disrupted in disease. Dynamical systems models are ones that describe the time rate of change of concentrations. Is it slow? Is it fast? Exactly at what rate does it occur? How does it occur? These things can be probed and measured experimentally, but it's hard and detailed information to get. So while, and fundamental physical chemical principles constrain these measurements: conservation of charge, conservation of mass, the very nature of how one molecule binds to another, what affinity of binding means. Fundamental physics of the problem constrains the form of these equations, but we can formulate. I've spent 25 years of my life doing this in cardiac myocytes. And so these are a different kind of model that are direct descriptions of what mechanism is. So what are their pluses? I think the big plus is this kind of function f that describes, I should have said, what do you do with these models? You numerically integrate these equations. You solve for x. You know their time rate of change. You can formulate that. You solve for the x. So you actually make predictions about how variables will change in time as you simulate the system. And that gives you a glimpse into the way the system behaves in its future evolution. Well, that function f, this so-called velocity field, captures deep knowledge about mechanism, this enormous amounts of mechanism built in there. I say these are the models we really want. We really do want to understand mechanism, but getting them is very, very slow, very hard, very challenging. Lots of experimental work, difficult, demanding work, hard to build these models. Computing them can be extremely hard as well. That's what I want to move to now. This is the cell that I've worked on for most of my life. It's a cardiac muscle cell. I want to understand how molecular networks and their perturbations lead to, in disease, lead to the generation of arrhythmias in the cardiac muscle cell and in muscle tissue in general. I've spent most of my life building these kinds of dynamical systems model of the cardiac myocyte. The details aren't important. What's important is there's just enormous amount of mechanism packed into these models. The way ion channels open and close and control the fundamental electrical excitation of these muscle cells, the transporters and exchangers in the membrane that are involved in regulating ion homeostasis within the cell, the way in which electrical depolarization of a muscle cell is coupled to calcium release that drives muscle contraction, the fundamental process of muscle contraction and force generation itself, mitochondrial energy production, force generation requires ATP, all active processes in a cell require ATP for fuel. These models describe how mitochondria produces ATP, signal transduction pathways that modulate all of this. Enormous amount of detail and machinery built into these models, and the models are now spatial. They're not one compartment, they're thousands of compartments, because I think arrhythmias are rare stochastic events. People can be born with mutations that predispose them to arrhythmia. If we take those mutations, study their nature, and implement them in deterministic models, these models will show an arrhythmia on every heartbeat, every single heartbeat. You couldn't live for 50 years if that was a representation of reality, but there are many people who live for decades with an inherited predisposition due to a mutation in a particular mechanism here, but it's a predisposition. And that's because I think fundamentally arrhythmias are stochastic. They're random. The right combination of factors modulates their probability of occurrence. And we need to understand how the parameters, the properties of these cells control the probability of occurrence of arrhythmias. To me, arrhythmia is a study of probabilities. That's what we want to know, how to model properties relate to the probabilities of arrhythmias. Now we can do it brute force, which is what I'm showing you here. I'm going to show you a simulation result where these random calcium release events that are abnormal are triggering an action potential in a cardiac myocyte at the incorrect time. You all know that the pacemaker of the heart is the sinus node. It generates a regular oscillatory rhythm that governs the regular contraction of the heart. It's these random calcium release events that can trigger abnormally timed action potentials that can generate an arrhythmia in the heart. So we want to understand how do properties of this model relate to the generation and the likelihood of generation of these arrhythmias in cardiac muscle cells. This is just a simulation. I think it's either this cell, I think it's number 2 here, is the cell in which there was an actual action potential triggered at an abnormal time. This is an arrhythmia, it can trigger an arrhythmia in the whole heart. Now, the challenge here is that it took us 20,000 core hours of simulation and crunching to simulate that for one parameter set. And we want to study hundreds, if not thousands, of parameter sets so that we can understand the relationship between choice of perturbation of a parameter and the probability of an arrhythmia. If we knew that, if we knew what controlled that probability, we'd fundamentally know what controls arrhythmia. And we'd be able to go into that model and say, oh, it's this ion channel here. And if we knew how to modulate its function to reduce that probability of arrhythmia, we'd have an antiarrhythmic drug. And so what do? The models, I like to say the models are becoming as complex as the living system. They really are, so much mechanism in them. Okay, so I've gone through the spiel of mechanistic modeling and machine learning methods. Now we're going to see a connection between the two. It's a forefront area, I think. Okay, so so what were the challenges? Building mechanistic models, hard to do, slow, difficult, plod along, but lots of mechanism. What's the strength and weaknesses of machine learning? Fast to do, no mechanism. What if we could bring these two together? What if there was a way of learning from this complex model a very simple representation that we could compute on quickly? And I mean, it's not, this simple representation can't possibly capture everything that's in this complex, much more complete model. But it might be able to capture in the narrow confines of a particular problem what we're interested in. And so, here's a focus question. If I were to choose a particular aspect of arrhythmia, a particular disease, and build a very detailed, complex model of it, can I find a way of boiling that super complex model down into the world's simplest model? The answer is yes. Here it is, again, our friend, the logistic regression model, believe it or not. It's one of the simplest statistical modeling techniques out there. So my student found a way of carefully working through parameter space in the big complex model so that we could do a small number of simulations of that model and build up this simple representation that told us how perturbations or changes to, in a small number, there's like 5 or 6 parameters here that we were focused on because we knew they were involved in this arrhythmia from other work, how they regulate the probability of arrhythmia. So we learned that from the data, tested it, the data, the simple model reproduced what the full-blown model did really, really well. Here's an extrapolation. Here's something useful that we did. We took the simple model. We looked at this genetic mutation called long QT syndrome 1. This is a family of mutations, a large number of different mutations that target a particular potassium ion channel in the heart that controls its electrical excitability. Great deal of work has been done on long QT syndrome 1. In this particular study, there were a group of experimentalists who took induced pluripotent stem cells from patients with long QT syndrome 1. These cells carry those mutations. They differentiated them into cardiac myocytes. They studied them and understood how this particular ion channel's function was modulated in the mutation. They did it quite exquisitely. We took that information and were able to make the following kind of prediction. And the prediction was, can we predict from these data, from the simplified modeling approach, the 30-year cardiac event rate associated with these mutations? This is the clinically measured lethality of those mutations, in essence. The answer is yes-ish, we can do that. I say yes-ish because the blue are the model predictions, the dots are the 30-year cardiac event rate for each of these 5 mutations. We don't get the absolute value right, but we get the relative lethality of the mutations right. We get their rank order right. So we know clearly that this mutation is less lethal than this one is less lethal than that one, and so on and so forth. So this is important information, comes from the world's simplest model. Here's my final meat slide, and it involves a bit of speculation where I think this modeling can go. This is a study that we're preparing now for publication. The way I think of this is that it relates to drug discovery, anti-arrhythmic drug discovery, but drug discovery in general. So anti-arrhythmic drugs typically are developed to target potassium channels, maybe calcium channels in the heart. And so I'm showing you a measure of activity of the potassium channel on this axis and a measure of activity of the calcium channel on this axis. These drugs bind to and either block or enhance this current, and they bind to and block or enhance this current. The question is, in this parameter space, if we knew the relative degree to which a drug blocks the calcium channel or enhances it, and blocks the potassium channel or enhances it, can we make an in silico prediction about the anti-arrhythmic nature of the drug? If we could do this, this is an adjunct to in silico drug development. We could make computational predictions about the way in which, the affinity with which a drug would bind to these channels. We could make in silico predictions about the percent block that could be achieved at a particular concentration of the drug. And if we can do this here, what I'm showing you here, we can make predictions about the overall effect of this drug on the probability of arrhythmia. And that's what we're doing here. And the only way that, the reason that we can do it is that machine learning model, the approach of learning a model that takes us from the complex to the simple. We can now do this with paper and pencil mathematics. It's not even a computational problem anymore. We can define, so suppose this is the operating point of the cell. This is the normal average degree of expression of this channel, and normal average degree of expression of this channel. That dot should have been lined up here. I misplaced it. Suppose that's our operating point. So this is our normal, so what this line is an iso probability of arrhythmia line. If we perturb away from normal, these, so for example, moving here, this reduction of the calcium current and the reduction of the potassium current yields the same likelihood of an arrhythmia probability that we normally see. But if we step on this side of the line or on this side of the line, things change. If we are pushed into this part of the plane, that means the drug is proarrhythmic. The probability of arrhythmia is going up. If we're pushed into this part of the plane, that means the drug is antiarrhythmic. Desirable. Not desirable, obviously. And so it is absolutely amazing to me how one can go from an unbelievably complex computational demanding model essentially to a pencil and paper model, where really important things like relative risk of arrhythmia or pro versus anti-arrhythmia can be computed with the pencil and paper model out of machine learning. So where do I think this is going? Cliff showed me this paper yesterday. He sent me this paper yesterday. I said, thank you, I'm putting it in my talk. Because it's MIT Technology Review. Look at that title. Whoops. Uh-oh, now I have to go backwards. There we go. Hundreds of AI tools have been built to catch COVID. None of them helped. You take a look at this paper. The essential message of the paper was these methods for predicting who's going to get COVID, who isn't, or what's their progression in COVID, whatever the prediction problem was. These methods didn't work well, and the question is why? And the answer that they're pushing towards in this paper is that the importance of what I call ground truth, taking you back to that second slide on what supervised machine learning is, the labels, assigning labels to features. I call that establishing ground truth. We know, you have to know what the label is, and you have to get it right. Remarkably, they were pointing out things like some of the chest images of lungs and their function that were used in this COVID study were actually from children, not adults, and children without COVID. How in the world did they get into the data sets? I don't know. But clearly this was not ground truth. And the point of the paper is that they didn't establish ground truth right. So creating ground truth is vitally important. I think there's another major machine learning problem that we have to confront. And I call it the out of distribution learning problem and the continual learning problem. So if we, in our work with Maine Health, learn an algorithm on data from Maine Health patients at Maine Medical Center, I bet you we take that algorithm to, I don't know, I'm just gonna, San Antonio, Texas. And they implement that algorithm as we published it, all the parameters, everything we did. I bet that algorithm won't work. We already know this for some prediction algorithms in heart research and heart disease, that at best you can do no better, and you're likely to do worse, and sometimes worse is to the point of failure. And why is that? It's that every hospital has a different area from which they draw patients. Patient demographics are different. I think that's the primary problem. He's just seeing different kinds of patients in different areas of the country, Just a second order, it may have to do with the equipment that's used to make measurements. In some cases, it can be related to how patients are actually treated in this hospital system versus another. But the essence of the problem is that at one hospital and versus another, the data distributions are different. And to continue with my main health analogy, if we only learn from data on main health patients, that may work for main health patients, hopefully it does, but it may not be transportable. So this is called the out of distribution learning problem. The continual learning problem is the idea that we have to deploy these algorithms in a way that they are continually learned from data and continually refined. I suspect this is the only way that we can deploy algorithms at sites around the country and the world, is if along with them we provide a means by which that algorithm can be retrained on the data from patients that that center sees. And finally, I know I've probably gone long. This is my last slide. So I think the other major challenge is potentially machine learning of dynamical systems models. Can we harness fast, powerful machine learning methods to actually learn these complex models from large data sets and accelerate all of that? That's what we attempted to do here. This was a study where we asked the following question. Patients with COVID are going onto ventilators. Ventilators are dangerous devices. They can cause lung injury. They can save the life of a patient, but they can also cause lung injury. And that's true in general. It's not only about COVID patients. So this is an important issue. If a patient has to go onto a ventilator, let's keep them on as long as they need to be on that ventilator and then get them off as fast as we can because they are dangerous devices. So the way we formulated that problem is, can we measure things in the patient that will inform us how to control the settings on a ventilator so that that patient reaches and stays at the target SpO2 level as quickly as possible? Can we learn a control law that tells us how to automatically adjust the ventilator settings, in other words? Ideally, to do this, we'd have a beautiful, complete mechanistic model of lung function and how blood gets oxygenated. We don't have that model. It's just an incredibly complex question. So we asked the question, can we learn that model from data? And so we tried to learn the model. We're another COVID failure. This didn't work out. It worked out for completely unanticipated, it didn't work out for unanticipated reasons. And the reason was, in order to learn this control law and build the model, we needed to know how ventilator settings altered the oxygenation level of blood. And as we got into the problem working on these COVID patient data sets, we learned, and you're gonna laugh at me, that the time stamps that were associated with ventilator setting changes were entered who knows when. They weren't read off the ventilator. They were entered when a caregiver had time to enter them. So it could be an hour, a 2 hour, 3, 4, 5, whatever it was, hours later. And so we couldn't cleanly identify at this moment of time, the ventilator setting was changed, it produced that effect, so it made it hard to learn the model. We really couldn't learn an adequate control model here. Now, we had our eyes opened, that's useful. We now know that we need to automate this data collection. We need to be able to get these precise data, and we learned how to go about building these machine learning constructed dynamical systems model. So that was all good, but ultimately it didn't work out. But in the long run, I think this is a very important area of research. So that's what I have. I hope it made sense. I'm happy to answer questions. Thank you for coming.
I think we have time for 1 or 2 questions, and I'll leave it to Rachel to tell us if there's anybody online asking questions. Here.
Ray, very nice talk. Your models only are as good as your data. So, and I have a couple questions and perhaps comments. The the septic shock model is taken from machine data, from heart rate, PO2, oxygen saturation, that sort of thing. Doesn't take into account biochemical changes.
All right.
And there are fantastic predictors of sepsis in biochemistry. Your last model with respect to the effect, how ventilators affect specific patients, you have a huge problem with biological variability. Everyone's different.
Yep.
It's very hard to develop a model when you have that kind of variability.
Right. So a couple of points. The sepsis model was actually, so the top features in the sepsis model, the number one feature, it's not going to surprise you, was lactate. So that's obviously a lab assay. That was the number one predictive feature. It's not measured continuously. It's measured infrequently. So one conclusion of the modeling was, we should really find technology that helps us monitor lactate more precisely and more continuously or more frequently, because that's the number one predictor of this transition. The other predictors, the other features that were important, what we learned was kind of surprising, that things like heart rate and blood pressures were not really the top scoring features. They were in the middle. And so getting this kind of insight from the data that we had, even in the presence of all of its limitations, I think is very useful. I think we only know what we can model, and I agree completely with the importance of being able to make precise measurements, of course. But we have the data that we have, and the best way to operate on that data is to operate on it and think about it in the context of a model. That's really my main message. The second thing was, I think your second comment was about the antiarrhythmic effects and variability, or
Just biological variability.
Yes, okay. So one thing I didn't say, I meant to say it, is that machine learning approach of building a logistic regression model out of a complex model, you get so much out of it. The kind of thing you're referring to is uncertainty analysis. If there is an important parameter that you only know with a certain confidence, that parameter is actually drawn from a distribution, or you can make reasonable assumptions about that distribution, its mean and its variance, for example, it's trivial to understand how that uncertainty propagates into what you're trying to predict. And it does so in a beautiful way. There's a lot to talk about there. There are examples where we've believed that mutations are insufficiently characterized in terms of the precision with which certain parameters are measured, that you can't make any inference about what those mutations will do regarding probability of arrhythmia. Because the distribution of the estimate of that probability is the uniform distribution, which is the maximum entropy distribution for a given mean. You essentially know nothing. And so there are very, very easy ways in that formulation to incorporate the notion of uncertainty and look at its effect. It's a very beautiful thing. It could be applied to any kind of noise in biological systems, imprecision in measurements, or variation in populations. The same technique could be used to look at all three classes of questions, I think.
Other questions?
Ray, really fascinating talk. Thank you. I'm curious, you mentioned there seems to be a little bit of a conundrum between needing clear data, accurate labels, and wanting algorithms to be able to be set up to be learned, to continue learning over time. Mm-hmm. Can you elaborate on how you would see developing that and what you would do to ensure that an algorithm doesn't go off the rails?
Yeah. So I'll give you my thoughts. They aren't going to guarantee that the algorithm won't go off the rails. We have to test that. It's very interesting. When we began the sepsis work, there was an internationally recognized definition of sepsis and septic shock that we applied to the data. to categorize a patient as being in sepsis or in septic shock. The strange thing we did was, since we had data coming in every minute, vital signs data every minute, we were able to assess minute by minute, what is our diagnosis for that patient? Are they in sepsis or are they in septic shock? Because we had to identify the patients who transitioned to septic shock so we could use them as examples to learn from. What we discovered was something new and very illuminating. Even from minute to minute, the diagnosis could differ. We were simply applying standard clinical criteria for separating patients with sepsis from those with septic shock. The assessment was fluctuating constantly over time. There was no stable state of diagnosis. It's literally as if a physician walks in, makes a diagnosis, leaves the room, comes back 5 minutes later, looks at the new data from the patient, they'd reach a different conclusion. That's how extreme it was. So I'm bringing this up because that clinical definition of sepsis and septic shock had a fundamental flaw in it, that nobody ever imagined that some nutcase would come along and apply that definition every minute to data from a patient. They wouldn't recognize what we came to call temporal instability. And so I'm making this point because I think we can only do the continual learning in areas of diagnosis where we're pretty darn confident that we know how to make the diagnosis, that experts can do it and generate the correct class labels. That lesson showed me that definition didn't work well. Subsequently, the definition was updated, and they corrected it wasn't a flaw, it was an aspect of the older definition that was causing this problem. And now the state labels became very, very stable over time. So they corrected their understanding of how to, but this is only going to work in problems where you have clear, accurate consensus definitions of what the patient's state is so that you can generate these class labels. Second, you're going to have to do computational phenotyping of patients on the fly. As new patients come in, we can't have teams of people looking at them and say, oh, they're an example of this, or they're an example of that. We need to be able to implement the diagnostic criteria computationally, know that they're stable in the sense that I just described, so that our automatically generated class labels are accurate. And then over time, we build up and accrue new patients with accurate class labels. So we can do one of two things. We can, over time, as we get new examples to learn from, we can rebuild the classifier. That's doable. Or we can figure out how moment to moment, data element to, data observation to data point, learn how to incrementally update learning algorithms. I think that's the more interesting thing. But I literally think this is what we need to do to support continual learning, and it will only work where there's a pretty solid understanding of how to diagnose A versus B. If there's ambiguity, you will never be able to publish the data because the reviewers will be all arguing you didn't do the diagnosis correctly. So
It is my job to wrap us up.
okay.
So I want to end by saying thank you to you, Ray. It is the primary thesis of the Roux Institute that computational thinking, data science, and data modeling will transform all of our lives in the next century. And there is no place where we believe that that will be most profound than medicine and life sciences. And so your leadership in this area and in this field, along with all of our partners from University of Maine and USM and IDEX and Maine Health and Northern Light Health and all the folks that we're working with to build this center of excellence in computational medicine here, it's really exciting to see the very beginning of it. And we thank you for your talk and for your leadership and for doing this here this evening. It is also my pleasure to invite you all to join us for some refreshments, ask more questions, get to know each other. And I'll just end by inviting you to the next two talks in the series. We have a lunch talk on May 11th. It's virtual with Melanie Torrey, whom Ray mentioned, who's going to be doing an introduction to the field of data visualization. And then we have our next in-person session here on Wednesday, May 25th, where a member of the faculty in our Institute for Experiential AI will be doing a talk on ethics in AI. And we welcome you to spread the word and come and join us again. So thank you all and have a lovely evening.